Hi, my name is Matt Jenkins, and I am a senior at the University of Tennessee, where I am a research assistant in Dr. Matthew Cooper's Behavioral Neuroscience Lab. Today I will be discussing how acute social defeat alters microglia activity and how it interacts with a subsequent immune challenge. Our lab researches social stress using Syrian hamsters as a model. We use Syrian hamsters because they are an aggressive and territorial species that naturally form hierarchies and dominant-subordinate relationships when housed together. Social stress is one of the most common stressors experienced by humans. 9 out of 10 people will experience trauma in their lifetime, but only 1 out of 10 go on to develop a stressor-related psychopathology such as anxiety, major depression, or PTSD. Studies have shown that some of these psychopathologies, such as major depression and PTSD, are associated with inflammation, and that artificially induced inflammation can result in depressive-like symptoms and social withdrawal, and that these effects can be reversed with antidepressants. The ventromedial prefrontal cortex is an evolutionarily conserved brain region in vertebrates that is thought to regulate an organism's interaction between its internal state and environment. It is connected to many other brain regions, including many limbic structures such as the amygdala, dorsal raphe nucleus, and hippocampus, where it provides top-down inhibitory control. The ventromedial prefrontal cortex is divided into the prelimbic and infralimbic cortices. Studies have shown that activity in the infralimbic cortex is required for the formation of an extinction memory, and that lesioning of the infralimbic cortex before environmental enrichment eliminates the effects of acute social defeat. As for the prelimbic cortex, the roles are mixed, but it has shown to drive development and expression of fear-related memories. Our lab has shown that dominant animals show increased activity during social defeat, and that inactivity of the ventromedial prefrontal cortex leads to stress vulnerability. Therefore, the ventromedial prefrontal cortex activity is critical for stress resilience. Microglia are the resident macrophages of the central nervous system. IBA1, or ionized calcium binding adapter molecule 1, is a protein specific to microglia and macrophages. Innate immune activity in the brain can lead to rapid proliferation and mobilization of microglia. These microglia can change their morphology as their pro-inflammatory activity increases, as you can see in the figure on this slide. When microglia are in a quiescent state, their morphology more closely matches the picture on the left of the figure, and as they become more active, they begin to appear more amoeboid, like the picture on the right of the figure. As they become more amoeboid, they start showing more pro-inflammatory activity, such as increased cytokine and reactive oxygen species generation and increased phagocytosis. IBA1 is expressed in every stage, but is upregulated as they increase their pro-inflammatory activity. Studies have shown that animals that proactively react to stress show fewer cytokines in the brain. And this top picture is actually a picture of a microglia from a hamster brain taken in the Cooper lab. Alright, so now for a brief microbiology lesson. Lipopolysaccharide is an endotoxin, meaning that it is a toxin that is present inside the cell and is not secreted. It is a component of gram-negative bacterial cell walls and is a potent immune activator. LPS binds to the toll-like receptor 4, which is a receptor present on certain immune cells, which then leads to NF-kappa B signaling, which is part of a pro-inflammatory cascade. Septic shock is the result of a massive pro-inflammatory cascade caused by lipopolysaccharide. The aim of this study was to determine whether acute social defeat alone or with systemic LPS leads to changes in microglia morphology and IBA1 expression, and whether or not these changes persist over time. It was hypothesized that acute social defeat would increase IBA1 expression in the ventromedial prefrontal cortex cells and would potentiate cellular responses to LPS. It was also hypothesized that IBA1 positive cells would exhibit changes in morphology consistent with activated microglia and that these changes would persist over time. This study was divided up into two parts, the inflammation experiment and the time course experiment. The inflammation experiment sought to determine whether acute social defeat alone or with systemic LPS leads to changes in microglia morphology. For this experiment, 58 adult male Syrian hamsters were divided into stress and no stress groups. The stress groups received acute social defeat, where the subjects were exposed to 3 5-minute encounters with a larger, more aggressive hamster in the cage of that larger hamster. 24 hours later, the animals were divided into 4 groups. One group received a saline injection, and the other 4 received various doses of lipopolysaccharide. One group received 20 micrograms per kilogram, one received 100 micrograms per kilogram, and the last group received 500 micrograms per kilogram of lipopolysaccharide. These animals were then left alone for 4 hours before their brains were collected. The time course experiment, which sought to determine whether changes in microglia morphology after acute social defeat persisted over time, was very similar to the first experiment, except none of the animals received lipopolysaccharide injections. 19 adult male Syrian hamsters were divided into stress and no stress groups, and again, the stress groups received acute social defeat. And then they were left alone for 7 days before their brains were collected. This is a figure showing real images of IBA1 stained microglia in the ventromedial prefrontal cortex from each of the groups in the inflammation experiment. As you can see, the IBA1 density is increased in the animals that received acute social defeat, and this increases as LPS dose increases, indicating that the microglia are in a more active state, referring back to the microglia activity chart. In figures B and C at the top of this picture, these show that optical density of IBA1 positive microglia is increased in both the prelimbic and infralimbic cortices of the ventromedial prefrontal cortex. Optical density is plotted on the y-axis, and the x-axis shows each of the LPS dose groups. Animals that received acute social defeat are represented by the white circles, and the non-defeated animals are represented by the black circles. Notably, the 20 microgram per kilogram group showed the biggest difference between stress and no stress animals. We also measured soma size of the microglia, which is just the cell body size. In figures D and E, soma size is plotted on the y-axis, and defeated animals are represented by the black triangles and no defeat by the white squares. We chose to compare the 20 microgram per kilogram dosage to the no defeat animals, as this seemed to induce the most IBA1 expression. We found that the soma size of IBA1 positive microglia in the infralimbic and prelimbic cortices of the ventromedial prefrontal cortex is increased in animals that received acute social defeat and 20 micrograms per kilogram of lipopolysaccharide compared to animals that received saline. On the bottom, figures F and G are similar to D and E, except that soma to cell size ratio is plotted on the Y. We found that IBA1 positive microglia in the infralimbic but not the prelimbic cortex of the ventromedial prefrontal cortex showed increased soma to cell size ratio compared to animals that received saline. And again, these findings are consistent with the idea that upregulation of IBA1 is associated with increased microglia activity. For the time course experiment, figures A and B show that the optical density of IBA1 positive microglia did not differ between animals that received acute social defeat and those that did not, in either the infralimbic or the prelimbic cortices of the ventromedial prefrontal cortex after 7 days. The soma size also did not differ between these groups. However, the soma to cell size ratio did differ between stress and no stress animals in both the infralimbic and prelimbic cortices of the ventromedial prefrontal cortex. These findings suggest that after 7 days, microglia do not show increased IBA1 expression, but they appear more amoeboid, which, referring back to the microglia activity diagram, correlates with increased activity. In conclusion, we found that acute social defeat can prime microglia in the infralimbic and prelimbic cortices of the ventromedial prefrontal cortex following subsequent LPS exposure. These findings are important because they indicate that acute stress can increase inflammation and can increase stress responses to future challenges. Therefore, inflammation could be a risk factor for stress-related psychopathologies. I would like to thank all of the people in the Cooper lab, specifically Dr. Alex Grizzel, as this research was part of his dissertation that earned him his doctorate's degree last year. Thank you for listening.